All right, we are now in part 3 of chapter 23, Evolution of Populations. So in the previous portion of the video, we talked about, the mechanisms that can alter allele frequency. We got done talking through and summarizing natural selection, and we have gone done and talked through genetic drift. How does that affect allele frequency? So last but not least, the third mechanism that can result in change of allele frequency is gene flow. What is gene flow? It is the transfer of alleles into or out of a population due to movement of fertile individuals or their gametes. So this type of gene flow tend to reduce genetic differences between population, and second, they affect how well populations are adapted to a local environment because you are blending, in a sense, the genes between population. That's why you end up with less genetic differences. And again, less genetic variations may lead to less likely to adapt. And that's why it can affect how they adapt to the environment. So for example, water snakes in the mainland versus islands of Lake Erie, as you can see in this picture here, differ in their banding coloration. So mainland water snakes usually are strongly banded, which is actually a dominant trait, and live in the marshes. Whereas the island water snakes are unbanded, which is a recessive trait, and they tend to live in rocky shorelines. So being unbanded in the islands help with being camouflaged from predators in those rocky shorelines. That's why they can live there. And thus unbanded snakes are favored by natural selection on the islands. Interestingly, we observe that not all water snakes on the island are unbanded because we just said unbanded. Being unbanded on the island is what favors its survival, being able to become a camouflage from predators. So why did natural selection not wipe out all banded snakes from the island population, since they are supposedly not favored in the island population? Well, it turns out that this is all due to gene flow, which means gene flow in this particular case from the mainland. Every year, about 3 to 10 water snakes from the mainland will actually swim to the island and join their population. These migrating snakes will then transfer their banded alleles to the island, which is what prevented natural selection from removing all banded alleles on the island. In a sense, gene has flow from the mainland onto the islands. In this particular case, it was the banded alleles that got introduced into the island population. That's what gene flow means. So in a sense, before we move on to the next section, this wraps up the 3 mechanisms that can lead to change in allele frequency, which may ultimately lead to evolution of species. So make sure you remember definition and example from these 3 mechanisms that alter allele frequency, leading to evolution. So the last portion of this chapter then focuses on different types of selection you can have under the big umbrella of natural selection. Remember, I mentioned that evolution by natural selection is only mechanism that is not random, because we talked about natural selection, genetic drift, and gene flow. Genetic drift is by chance alone. You cannot predict. And then your gene flow is also kind of random. Natural selection is the only one that consistently increases allele frequency that provide a reproductive advantage, which means this is what produces what we call adaptive evolution. So how does natural selection cause adaptive evolution? Well, first, certain traits can lead to greater relative fitness. We're not talking about fitness in a sense going to the gym type of deal, but we're talking about traits within a population to provide the species from surviving in a particular environment. So this relative fitness contributes an individual mix to the gene pool of the next generation compared to other individuals. So for example, the snake jaws enable swallowing prey that are much larger than their own head, as you can see in this picture here. Snakes have known to be open their jaws so wide that they can swallow things bigger than its head. So that's what relative, in a sense, this ability to ingest huge things is a relative fitness that allowed this particular trait of snake to prevail. But it is not direct competitive contest among individuals. It only increase favorable traits to improve the environment. Meaning that is not like let's have a competition to see who is the strongest here. We're talking about it is those that are more favorable to improve life in a particular environment that results in this particular trait being favored, okay? Not because there's a competition amongst the individual, because the textbook says that this is usually the misconception of why it's not been mentioning natural selection as survival of the fittest, which you usually commonly hear, because they think is not a really scientifically correct of saying what natural selection is not a competition. It is just this particular animal or species or part of a population just happen to have this trait that is better suited for this environment survival. If the environment changes, they can change its survival rate. So that's why it's not a competition. And selection actually acts more directly on the phenotype versus genotype. Why? Because different genotypes can sometimes result in the same phenotype. For example, if we refer back to the purely homozygous dominant and heterozygous situation, purple flower and white flower, or yellow seeds and green seeds in Mendelian genetics, even though their genotypes are different, if we're talking about heterozygous, their phenotype are still the same. Whether you have, for example, a big A, or a big A, little a, this one and this one phenotype are the same in a purely homozygous recessive situation. So that's why selection acts more on the phenotype and not the genotype, because evolution will not see, oh, this is a homozygous, this is a heterozygous, I would prefer heterozygous. It's not like that. Evolution is just natural selection is just based on traits that are better for survival, which is why it is more so on the phenotype versus the genotype. All right, and it depends on which phenotypes are favored for the environment. Natural selection can actually alter frequency of distribution of heritable traits in three ways. Either you have directional selection, disruptive selection, or stabilizing selection. What are these three things? Well, directional selection means that it occurs when conditions favor individuals with one extreme of phenotypic range. Remember, this is only one extreme of a phenotypic range. So this is common usually when a population's environment changes or if that population migrates to a new habitat. For example, the finches we talked about from the Galapagos Islands beak increase in size when a drought happened. So that's an environmental change that happened in this area, and hence the finch's beak all kind of go in one direction to big, larger beak size. Disruptive selection occurs when conditions favor individuals at both extremes. Keyword there, both extremes of a phenotypic range over those with intermediate phenotypes, which means it is the two extremes that are favored. If there's something in the middle, it is not favored. If we go back to our wildflower populations case, let's say that red flowers and white flowers are somehow favored in this particular environment. Pink ones are not. That's what we call disruptive selection. Another example is black belly seacracker finches. They actually display two distinct beak sizes. Either you have very small beaks for soft seeds, or very large beaks for hard seeds. And that turns out that those with intermediate beaks actually can't crack either seeds. They can't very efficiently crack anyway. They may be able to do it, but it's not very efficient compared to either small or big. So that's why it kind of select out your intermediate phenotype. So disruptive selection favors survival of both your extremes of the traits, while those intermediate ones will die off. I will show actually graphs of these three different selections in the next slide so you can better visualize what I mean, because I think sometimes words are like, what? So there are pictures that talk about these three different types of selection. I'll get to that in a bit in the next slide. So third selection is called stabilizing selection, which actually is the opposite of disruptive. It favors intermediate variants over the extreme phenotypes. Now, in this case, because it favors intermediate variants, it actually reduces variation, which is why it stabilizes a particular population. So, for example, birth weights for human babies usually average anywhere from 6.6 to 8.8 pounds. With much smaller babies or much larger babies have less chance of survival. So it actually favored those in this average range, which is the intermediate variants. So again, in a sense, remember, your stabilizing selection is the opposite of disruptive selection. It only favors survival of the intermediates. So if you didn't understand what I mean by words from the previous slide, here are the graphical depiction of the three selections. Here is the graph showing you the original population with mouse that varies from very pale white color to a little bit tan to brown, dark brown, and really dark color mouse. So it's a whole range of mouse fur color. Okay, so this is the original population with peak being your intermediate or sort of an even distribution of these different phenotypes, with the middle color being the most prevalent in this particular population. So I see here in directional selection, which is the first type we talked about, it only goes one way, which means it results in the evolved population either going to the left or to the right of the original population, depending which of the extreme phenotypes the environment favors. In this case, the curve shifted to the right, which means it favors darker fur mouse. So those that were lighter color that were here have died off. Maybe it's a survival kind of thing where the dark mouse was able to hide or camouflage themselves better in the environment. That's why it shifted in one direction. So that's directional selection. Remember, directional with the curve shifting in one direction. Then you have second graph here that depicts disruptive selection, where the evolved population moved to both the right and left side of the original population. Both extreme phenotypes are favored. That's why you have this kind of like camel hump curve right there, okay, in this particular picture. Excuse me. So that's why it's not shifting the peak to one way or the other, but it had kind of divided into two peaks, moving to the right and moving to the left. And then the last third graph here depicts stabilizing selection, which is where evolved population moved to the center of the graph, even more so in this particular case, favoring only intermediate phenotypes. So maybe there was a wider distribution between these colors, such that the kind of light tan color and the darker brown color was also prevalent. But in stabilizing selection, it only favors that middle brownish color mouse. So it kind of gets even thinner in that intermediate phenotype. So that's why it's called stabilizing selection, minimizing variation. Now you just have one variation of fur color in this particular mouse population. So in addition to natural selection, there is also something called sexual selection, which is a process where individuals with certain inherited traits are more likely to obtain mates than the others. So this type of selection drives what's called sexual dimorphism, which is where there is a difference in the secondary sexual traits, such as different size, different color, or even different behavior between males and females of the same species that allows one to prefer one over the other, or one of the males or one of the females over the other. So how does sexual selection works? There are two ways. One is called intrasexual selection, which means individuals of one sex compete directly for males or mates of the opposite sex. So for example, you have patrolling male that kind of defend the status of by defeating smaller, weaker males in combat. That is, kind of typical in guerrilla packs, where there is one patrolling male in the guerrilla pack, and they would defeat smaller, weaker ones. So to kind of defend their status and make sure they're always the lead male in that particular group. Second way is called intersexual selection, which is talking about mate choice. Individuals of one sex, turns out that usually it's females, are more choosy in selecting their mates from the other sex. So as you see a lot of times in the wild animal population, you will see males of a particular species looking brighter in color, prettier, more beautiful, and that is because of, the whole purpose of that is for them to attract mates. In this case, attractive males will attract females to mate with them. So their choice depends on showiness of the male's appearance or even sometimes behavior. Showy traits, actually, interestingly, may not be adaptive for survival and may even pose greater risk. Now, this is interesting. They may not be adaptive and may even pose risk to their lives. For example, brighter colors in males may actually make them more visible to predators. So this means that it is more important to take the risk in having brighter, flashier colors to attract mates and produce the next generation of offspring than it is to protect their own safety. That's really what it means. So if the animal can only survive but live alone and can't reproduce, then it could be the end of that species. In order for that population and species to live, they have to reproduce. So if they don't reproduce, that can lead to extinction. So remember, we have repeated this multiple times, that natural selection is both the ability to survive and reproduce. So that's why in this particular case, they would sacrifice their survival for the reproduction, the higher rate of reproduction, by having these flashier colors, or in behavior's case, it can be louder voice compared to other males in the population. So in addition, usually these secondary sexual traits are correlated with mates having better genetic quality that increases the species survival rates. Now this is pretty interesting. When we're talking about, let's say, just like in this picture here, a female peacock preferring brighter color male peacock, it's not like that female peacock can see, oh, that's a male with higher survival rate offspring. Okay, they don't know that, but they just know that this male has very pretty colors, maybe even behavior that's separates them from the other males. So research studies have shown that those males with more showy colors or better mating calls or rituals that attract mates not only have an easier time finding a mate, but also their offspring tend to have better chances of survival, leading to the conclusion that female mate choice can be based on a trait that indicates whether male has good genes, quote unquote. So it has shown in animal population that this is true. So the last type of selection discussed in this chapter is called balancing selection, which is a selection that preserves allele variation by maintaining two or more phenotypes in a population, which means not all unfavorable alleles are eliminated. It doesn't cause 100% frequency in the favorable alleles, is what this means. So this balancing type of selection can be a result of two things. One is called frequency-dependent selection, which means it is the fitness of the phenotype that depends on how common it is in the population. how common it is in the population, not necessarily the case that is better adapted to the environment. So for example, scale-eating fish actually maintain frequency of left mouth versus right mouth phenotypes close to 50-50, okay, which means 50% of the population are left mouth, 50% of the population are right mouth, well, for these allele frequencies, okay, so we're comparing left mouth versus right mouth as our P and Q, and they tend to be 50. It depends on which prey is more common that year. So see here in this particular picture, we have these gray fishes right here, which are the scale-eating fish. And of course, they're named as scale-eating fish because they literally eat the scales off their prey. So the orange one right here is their prey. So when right mouth fish is common, it actually, the scale-eating fish is common, its prey will guard against attack from them. So left mouth fish then have an increased chance to eat its prey because the prey tend to gonna guard the right side because they know, oh, right mouth fish is common this year or this season, so I'm gonna protect my right side more. So left mouth fish then will have an increased chance to eat its prey in the next season. So this can change from year to year, which causes the frequency of left mouth and right mouth fish to oscillate over time, as you can see here in this graph. One year it is, so this is frequency of left mouth individuals. Left mouth individual prevails. Next year, it kind of goes down. Next year, it goes up. Next year, it goes down. So this is why it oscillates depending on this prey. So second way we can end up with balancing selection is when there is a heterozygous advantage, which means it leads to maintenance of two or more alleles of a particular locus because heterozygous by genotype, not phenotype this time. Before we were talking about phenotype, but in the stabilizing selection. But this time, when we're talking about heterozygote advantage, it is by genotype heterozygous, exhibit greater fitness than both homozygotes. So for example, even though sickle cell disease is seen as a negative disorder, as we talked about earlier, one might think that even being a carrier for the heterozygote, as a heterozygote for the disease is a bad thing. But it turns out that when one is heterozygous, it actually is, or heterozygous for sickle cell disease, it actually is a less severe form of the disease. That's one benefit. Second, it protects, which is really surprising, it actually protects against most severe effects of malaria, which malaria is a parasite that infects red blood cell and kill them. You can die from malaria because it kills off all your red blood cells and you can't have enough oxygen delivered to your body, and that's how you die. So these, note that heterozygous advantage, again, is that you have the genotype that is the matter for the population, not the phenotype. If the phenotype of a heterozygous is an intermediate to the phenotypes of both homozygotes, then the heterozygous advantage is a form of stabilizing selection. Now, if it was a pure dominant recessive type of genetic pattern, inheritance pattern, where heterozygotes takes the dominant phenotype, not an intermediate, then it is not a stabilizing selection. So whether a balancing selection is equivalent or heterozygous advantage is equivalent to a stabilizing selection, all depends on what the heterozygote status is. In this case, you have three different phenotypes, which is homozygous for sickle cell, you have very diseased individual, and then you have heterozygote for sickle cell disease, which actually provide the heterozygous advantage because it protects against malaria. They say that because the sickle cell is kind of faster to be gone, in a sense, they will reproduce red blood cells more often, which is why they protect the individual against malaria, because that's where they infect. Is the red blood cells. So if these malaria are living in the sickle cell, they actually get killed off faster, just because of the intrinsic function or trait that sickle cell is more often killed faster by your normal red blood cell. And then you have, of course, homozygous normal individuals who are not, doesn't have the disease at all. So lastly, to conclude this chapter, we must be clear that although natural selection, excuse me for my change in tone. So natural selection leads to adaptation. It does not result in what we call perfect organism. It does not result in that, meaning it does not result in an organism that is perfectly fit for their environment that will make them invincible. There is no such thing. There's no such organism because of the four following reasons. First, selection can only act on existing variation. Existing is the key word there. Let's say ideal perfect traits may not actually be in the existing variation available in the population. For example, these picture here is depicting what's called the snowshoe hares, which is a type of rabbit that are able to camouflage against predators by changing their coat color that mimics snow. That's why you see a kind of brown tan color rabbit here or hair here versus a white color rabbit bunny here. So this white color is supposed to mimic the snow. So when it's time for winter to come, the rabbit will change their fur color to better camouflage themselves against the snow. Well, climate change actually led to a later snowfall. So winter has been delayed since climate change happened, but the rabbit's change in coat color was not, relating to our climate change now. It doesn't adapt to our climate change. So that's why the timing of color change remained the same. And this change of adapting to color, the color change adapting to the climate is not an existing variation. If it was, then it would have been perfect. Like they would change to white color, let's say according to different temperature, for example. When it gets cold, that's when it changes the coat color. Then that would be a perfect variation to have, and they would not be impacted by the climate change. But in this case, they were impacted because of the fact that their coat color change was not a temperature affected thing. It's a time affected thing. And that's why they will be actually not camouflage with climate after climate change happened, and they will appear white when everything is still not covered by snow yet, which in a sense actually makes them more obvious to predators. Second, evolution is limited by historical constraints, which means evolution cannot or does not erase existing structures and build and design new complex structure from scratch. It's not erase the board and let's draw again, okay? It operates on traits an organism already has. For example, we humans, it would be nice if we can fly. We can, have an advantage over a lot of things. But humans can't just grow wings all of a sudden because it is not an existing structure or trait that we already have. We cannot let's erase the arm bones and then now we can redesign our arm bones to look like wings. That's not how it works. Now, third is that adaptation are often compromises, which means is not a perfect match between adaptation and the traits or variation that is already existent in the population. Some organisms may actually face many trade-offs where ability to perform one function may reduce the ability to perform another. So for example, seals may walk faster or better if they have legs instead of flippers. But if they have legs, they won't swim as fast as they can. So that's why this is a trade-off. They have flippers that make them harder to walk on land, but they actually allow for them to swim faster in the water. Fourth is chance, natural selection, and environment. All of these things interact. Chance events affect subsequent evolutionary history of population. So for example, storm wind that blows organism randomly to another location, kind of like what we talked about with genetic drift. And then environment can also change from year to year. So just because you have this perfect organism in this environment, well, what if that environment changed the next year? Kind of like that drought situation with the finches. Well, then that organism was not fitted anymore. And let's say the environment changed back to favoring soft seeds, okay, soft, which then the small beak would then be favored in the finch's case. So it can change yearly, the environment, which means you cannot create or result, natural selection does not create a perfect organism. It's just selecting for the trait that best survive in the given environment with these four, according to these four constraints, okay? All right, so that is the end of the chapter. We will now go over some practice questions from the textbook and to give you some, see if you understand the basics. So in evolutionary terms, an organism's relative fitness is measured by its, which choice? B, contribution to the gene pool of the next generation. Next question, red short-horn cattle are homozygous for the red allele, white cattle are homozygous for the white allele, and roan cattle, which is usually somewhere in between the red and the white, roan cattle are heterozygotes. So population A consists of 36% red, 16% white, and 48% roan. What are the allele frequencies then? So let's assume, because they only gave us percentage here, so let's assume there are 100 cattle in this population. And then if there's 100 cattle, that is times 2, equivalent to 200 total alleles in this particular population. So let's say P is equal to red, Q is equal to white. And so you have P being red alleles, you have 36 that are red, and then times 2 for the 2 alleles, which makes them red. Let's say it was RR or something, maybe gives them red. So that is your 72. And then your, let's say, roan is heterozygote. So 16 of those, or 48 of those are then your roan color. Okay, so 72 plus 48 added together is then what gives you 120. Okay, so 120 of the alleles will be big R, given in this situation, divided by 200 total alleles. That will give you 0.6 as your allele frequency for red. And then we know that 1 minus P is equal to Q because P plus Q should equal to 1. So Q then should equal to 0.4. Okay, so that's why the answer is red is 0.6, white is 0.4 in their allele frequencies. Indicate which of the Hardy-Weinberg condition is being violated in this example. Some moths on a tree are easier to see due to their lighter color, therefore eaten by predators more often. Which is it? It would be no selection. Those are the variation in colors allowing some moths to survive more easily than others, therefore selection is occurring. Because remember, no natural selection should happen for Hardy-Weinberg equilibrium. So indicate which of the Hardy-Weinberg condition is being violated in this example out of the 5. Male elephant seals show aggression towards other males, resulting in dominant males mating with several females and other males mating with few or no females. Which is it? It should be random mating occurring. It is not random. There is a pattern to the mating going on that is described here, so it makes non-random mating, which violates the Hardy-Weinberg condition. Again, indicate which of the Hardy-Weinberg condition is being violated. Due to global warming, a river has dried up, allowing 2 different rabbit populations that were previously isolated to mate with one another. Which of the following is it? No gene flow. So when the river was there, the species were isolated, no gene flow was occurring. Now that gene flow can happen because you have now one is able to mate with the other, this can reduce differences between the population, which is gene flow is happening now in this case. The textbook discusses how the conversion of quality habitat into farmland has greatly depleted the greater prairie chicken population, and as a result, their genetic diversity. That's why a lot of them died off. So which of the following occurred in this particular example? bottleneck effect because there was a drastic drop in population size, and genetic diversity is causing the greater prairie chicken to undergo a genetic bottleneck. Indicate what type of selection is occurring in this example. Due to less snowfall in the area, white mice are predated on more than those are the intermediate or dark colored mice. It is directional selection. The population is shifting towards darker colors, moving the bell curve over to the right as we have discussed in lecture. Indicate what type of selection is occurring in this example. During a drought, it was discovered that finches with large beaks and those with small beaks were more successful due to food sources available during the drought. So both large and small. So it should be disruptive selection. In this example, the extremes are being favored and the intermediates are not being favored. So here are the calculation ones coming. So the frequency of cystic fibrosis, a recessive genetic disease, is 1 per 2,500 births among Northern Europeans. Assuming random mating, what is the frequency of carriers? So here we have 1 out of 25,000 births that have cystic fibrosis. That is equal to 0.0004. And then that is actually your Q squared because this is a recessive genetic disease. So Q, which then would be the square root of that, is equal to 0.02. And remember that P is equal to 1 minus Q, which is P then is equal to 0.98. And so 2 for carriers, it is representing your 2pq for the Hardy-Weinberg equation. So 2pq is equals to 2 times 0.02 times 0.98, which is then equal to 0.04 or 4% of the population. So it is 1 over 25 or about 4%. Which of the following evolutionary mechanism increases the amount of genetic variation in a population? It is mutation. Remember, the creation of new alleles via mutations and genes via gene duplications are the mechanisms by which genetic variation of a population can be increased. So genetic drift and all forms of natural and sexual selection tend to actually decrease genetic diversity. Which of the following is required in order for evolution to occur? Answer should be heritable variation. You have to be heritable trait. We already discussed that. Non-heritable traits should not affect the next generation of offspring, hence no evolution should occur. Which means all the other ones is then not required for evolution to occur. So which of the following can affect genetic variation within a population? All of the above. So in order for to have genetic variation, all of these things will affect it, whether increase or decrease. Now listed below are genotypic frequencies for 4 populations at the W locus or the gene. So which population or populations, if any, are in Hardy-Weinberg equilibrium? D, 0.25 for WW, 0.5 for heterozygous, WW, homozygous dominance, and then 0.50 for heterozygous and 0.25 for homozygous recessive. So the fact that WW is equal to P squared, because P squared is equivalent to the genotype for homozygous dominance. So P squared is equal to 0.25. Square root of that would be the P, which is 0.5. And then Q squared, which is little v, little w, is equal to 0.25. Again, square root of that would give you Q, which is 0.5. Then 0.5 plus 0.5 is equal to 1, which means this population is in Hardy-Weinberg equilibrium because it has to equal to 1. Whereas if you do the same for any of these others, you will not have 1 as a result. So listed below are the genotypic frequencies for 4 populations at the H locus or H gene. Which population, if any, are in Hardy-Weinberg equilibrium? Turns out that all of them are in Hardy-Weinberg equilibrium. So we'll do one of these as an example. Let's take this first one as an example. HH is equal to P squared, little h is equal to Q squared. So P squared is equal to 0.36. Again, P then is equal to square root of that, which is 0.6. And then you have Q squared equals to 0.16, square root of that. Q is equal to 0.4. 0.6 plus 0.4 is equal to 1. So it is in Hardy-Weinberg equilibrium. If you do so for the rest of these, you will find that they are also all equal to 1. So all of them are in Hardy-Weinberg equilibrium. So the following question pertains to the following scenario. One way to test whether evolution is occurring in a population is to compare the observed genotype frequency at a locus with those expected for non-evolving population based on the Hardy-Weinberg equation. In this exercise, you'll test whether the soybean population is evolving at a locus with two alleles, CG and CY, that affect chlorophyll production and hence their leaf color, because the more chlorophyll you have, the more green the plant will be. So students planted soybean seeds and then counted the number of seedlings of each genotype at day 7 and again at day 21. Seedlings of each genotype could be distinguished visually because CG and CY alleles show incomplete dominance, which means if you are homozygous for CG, their seedlings will have green leaves. If you're heterozygous, you have green-yellow leaves. If you're homozygous for CY, seedlings would have completely yellow leaves. So using day 7 data, what is the frequency of the CG allele P? Just the day 7 data. It should be A. So this is your day 7 data. 49 of them are homozygous, so that's why you multiply it by 2. 111 of them are heterozygous, so you add 111. And of course, there are no CG in your homozygous CY, so that is equivalent. So adding these two together, divided by the total is 432, because 260 individual, you have to multiply that by 2 for total alleles. So don't forget to do that. It can be easily mistaken. So that's why it's divided by 432, not 216, because 216 is the amount of individual organisms, but we're talking about allele frequency at this point. So total alleles of 216 individuals is 432 alleles. Okay, next, using day 7 data, what is then the frequency of CY allele, which is the Q? So CY is equals to Q, which is equal to, again, we're looking at day 7 data. You have 111 that are heterozygous for CY plus 56 that are homozygous for CY, which means you have to multiply those by 2. So then that is equivalent to 223 divided by 432, which is then 0.516. And that is why it is C as your answer. What is the expected Hardy-Weinberg equilibrium genotype frequency of CG genotype at day 7? Again, we're looking at your day 7 values. So it will be P squared, and we already calculated that P should be 0.484, so you multiply those by 2, you get 0.234. That's how often you will see CG homozygous, or 23.4% is really what it is. Calculate the observed frequency for the genotype CG at day 7. So CG, which is, again, at day 7. Genotype frequency. So 49 divided by 216 individual, because this is 49 CG, and you have 216 individual is 0.227, because we're talking about genotype frequency, not allele frequency. Compare the observed genotype frequency you just calculated with the genotype frequency expected if the population is Hardy-Weinberg equilibrium. At day 7, does it appear that the seedling population is in Hardy-Weinberg equilibrium, or is the evolution occurring? Again, feel free to pause the video, do your own calculation, and then come unpause it to reveal the answer. That's what, knows whether you understand it or not. So it should be C, population appears to be in Hardy-Weinberg equilibrium, okay? Because all of the allele frequency should be equal to 1. Or you can see here the observed, in this case, we're just comparing observed with the expected. So this is pretty close. I mean, there's never a perfect scenario where observed frequency is exactly the same as your expected frequency, but that's close enough. So we say it is in Hardy-Weinberg equilibrium. Okay, compare the genotype frequency at day 21 to the expected frequency and observed frequency at day 7. What appears to be occurring? So here's day 7, here's day 21, and their observed frequency. What should be happening? The population appears to be evolving, and there appears to be selection against genotype C. Why? Because this has somewhat about stayed the same. This has increased quite a bit, or increased a little bit, increased quite a bit, and then this one has actually decreased in the amount of frequency. So that's why it's selecting against CY. Homozygous CY individuals cannot produce chlorophyll. So that's why they have yellow leaves, as we've talked about. The ability to photosynthesize becomes more critical as seedlings age and begin to exhaust the supply of food that was stored in the seed from which they emerge. So what hypothesis could explain the data for day 7 and 21? Why is it selecting against CY? It should be D. At day 7, CY individuals were not being selected against because they had not used up the supply of food that was stored in the seed yet. But by day 21, they ran out of stored food, and now the plants are not surviving. So how do we expect frequency of CG and CY alleles to change beyond day 21? Probably what? Frequency of CY alleles will continue to decrease because now you really have deplete all of the seeds supply. Most of them will, all of them will, if anything. And then the frequency of CG allele will increase. And that is all for chapter 23, Evolution of Populations.